we'll explain a little why we think this is a relevant tool, very, very important tool for this type of disease that we want to work. So as a very introductory slide about the disease, it's slightly different, but we're still talking about leukemia. This is a chronic lymphocytic lymphoproliferative disorder, which is mostly characterized by the clonal expansion of the B cells. It's the most common leukemia in the Western world, very rare in Asia though. And it has a high degree of variability in the disease course. You have patients that they do extremely well, while there are another group of patients that they die in the first 1 or 2 years. And there are many steps. One of the big advantages that we have here is, again, it's a liquid tumor, right? We can take multiple samples. This is a MV of many people working solid tumors. So we have multiple samples. We can go to sequential analysis in good detail. We have millions of cells. And also, because of that, we have a very good representation of all the phases of the disease. And in many of those steps, going from a pre-malignant phase to the malignant transformation, going from the asymptomatic to a more progressive disease, and after relapse or refractory of treatment, we have key questions, biological questions. Some of those are related with the classification. So we have this pre-malignant condition called monoclonal B-cell lymphocytosis, which is basically, the definition is quite arbitrary. It's based on the number of cells. And one of the major interests that we have, we know that some of these patients remain stable for decades without developing the disease. Well, there is another 1 to 2% per year that evolve and they can become very aggressive disease. The reasons of that, they are not well established yet, and there are a lot of effort trying to identify which are the biomarkers associated with that, but most likely there is a genetic component there. And the same, once they advance, you have a watch and wait, that you don't treat the patient until they become progressive. And this is another key region that CLL is one of those diseases that has been a revolution in the number of type of treatments in the last few years with all these type of inhibitors, but based on the experience and again, going to the Darwinian principles, we know that in most of the cases, those cells will escape, will generate resistance, and most likely through genetic mechanisms. Like any other cancer, we know in the last few years has been an explosion. There are more than 1,000 exomes already sequenced through many different efforts. And now we have a very good understanding of the genomic landscape of the disease. Probably everybody saw this paper when we compared the type of complexity of the different diseases, from melanoma to some ALL, etc. So the CLL is in the left side, is in one of the less complex compared with some of the solid tumors. But still, when you see the genomic landscape, it's quite complex. So this can kind of summarize the most relevant driver genes that we see mutated in the disease, basically in all 8 different groups. But the interesting fact here is none of those is pathognomonic, or not even found in more than 15% of patients. And we have all series of combination of these mutations in different patients. Some of those, there are association with clinical variants. We know they are associated with outcome, but in most of those, we still don't understand what is the effect of such genes. And this is kind of with the same concept of Koichi, that by using all those tools, and especially by next generation sequencing, we have very powerful tools to get a very good understanding of the phylogeny and the clonal evolution, which is very similar of the Darwinian concepts. Following those concepts, and this is something that we try to understand, and we try to use single cell technology to better understand that evolution. With that in mind, we start with a pilot study where we select 5 individuals, 9 samples of those, because we did sequential studies. And we pick on purpose as a proof of principle to test a very complex cases like the one I just showed. Mostly the ones that we already have bulk sequencing and we know that they are a combination of mutations or they show parallel evolution. Other thing that was interesting to test was to go to our freezers and go to frozen PBMCs that have been there some over a decade and see if those were reliable to be used for this technology. The way that we used the Tapestri, and we picked this 34 gene panel that they have available, which actually include most of the putative driver genes that we see in CLL. So we took for this one, and in the samples, as I said, we look for samples like in this one, where this is by bulk tumor, but we see the variant reads in the first time point versus second time point. So you see an increase in multiple different mutations, but you don't know the phylogeny or the relationship between those. So we picked some of those cases previously analyzed, and the only thing that we know is that higher genetic complexity has been associated with a poor prognosis and shorter time to treatment. So this is the data, and we did this in collaboration with Mission Bio, sequenced the samples. So were used already the new sequencers. The number of cells that were used were between 5,000 and almost 13,000 cells. The coverage varies, especially depending on the sequencer, but one very interesting thing that we saw was the uniformity. I think that we were very pleased to see that overall, and my colleagues show in detail, but that was another panel, but that overall has a very good uniformity, so in most of the amplicons are covered in enough coverage. This looks very alike the other panel as well. We look for the correlations of the bulk versus single cell variant allelic frequencies that we characterize, and we didn't separate between, but in this cancer, most of them are SMVs, and it shows a very good correlation. And again, because we look for try to stress the system, we went especially for the ones that we have very small percentage of tumor cells or of the cells mutated. So going to cases, so I will show 3 of those cases. One is initially by bulk, is one of those extreme examples that we have many different clones, and that's something beyond, is something of our interest, is how to analyze those cases that they have this combination of mutations that in this case is a bulk, but you can see different activating mutations or in the hotspot of BRAF. If you go to detail, you see that the amount of different mutations that you have in the panel. So this is beyond this project, but one of the interesting things is why you have a particular gene like BRAF that is mutated in only 2 to 3% of patients, and in a particular patient, you have up to 10 different mutations. But what we try to do is, this is the percentage that we identify the bulk tumor, the percentage, and again, our threshold is in 1%, and we were very pleased to see that all those mutations were detected, not only detected, but also in a very good correlation in the percentage of detection, and even 2 that were below 1%, they were not identified by our algorithm for bulk, they were also identified here. What we try to do next is by running and separating, and they show clearly that all those subclones are observed there. And we prove that with a sequential sample where we see the same pattern. So also we try to do the first effort, trying to reconstruct the tumor phylogeny in cases, especially we focus in the pre-malignant condition going to evolve into CLL. So this is the first combination. Again, in this particular case, we have 3 different ATM mutations and 1 P53. The first thing that we did is, and we have 2 different time points, all were observed in second time point by both techniques, but even by single cell, again, we were pleased to identify 2 that were below detection with our bulk pipeline or the pipeline for detection in bulk. But besides detecting, we were able to start building maps based on that. And the interesting thing is, in the same patient, looks like based on and we're using the algorithms from the company, but we're able to see that we have an ATM a nonsense mutation where 3 independent subclones are generated, 1 with loss of heterozygosity in that particular one, but we have 2 additional subclones where they have the inactivation of the second copy of the gene. And more interestingly as well is we have a p53 mutation, but it's independent, it's not related to that particular clone. Another example, and if you recall, I talked about this is the scenario that we usually see, and the bulk sequencing is, okay, those are all the mutations that we got in this particular case, going from time point 1 and emerge in time point 2. But with this type of approach, we were able to start looking at different combinations and start making the puzzle, trying to understand a little more what are the association. And it's something that is repeated in the previous presentation as well, will show that how different subclones, they opt for this, sometimes for the same mechanism. Like this case, you have independent activation of KRAS in 2 independent subclones. So this is what we have at this point, but it looks like it's very promising. So we can say at this point that all the bulk sequencing variants were detected with this platform. We were able to do it. The sensitive was also, we identified mutations that were in less than 1% in frequency in the bulk tumor. There is a good correlation, very good correlation between the variant allelic frequency between bulk with the single cell across all the profile cases. In some cases, we start looking and trying to resolve the clonal architecture using this technology. And definitely, we are looking forward after this proof of principle to start focusing in larger scale in looking for the disease progression, and also trying before and after therapy with the new compounds that I previously cited. As we had the same observation of the other colleague, there are some of those amplicons they are not covering, or they are underperforming compared with the other ones, and those are the ones that need to be tweaked, and we try to work with that, or even generating a custom panel. And this is the people that has been involved in Mayo and Mayo institution, but also I have to thank a lot all the people from Mission Bio that have helped with the data generation and with the input in bioinformatics. Thank you so much.